Welcome to the Theoretical Neuroscience Podcast. I am the host, Etay Hay. Numerous neural models have been made over the last decades, but most of them can be called single-purpose models in the sense that they are made to address a single scientific question. In contrast, multipurpose neural models are made to be applicable for use in many studies addressing different scientific questions. In 2011, today's case, Etay Hay, together with colleagues in Israel and Switzerland, published a seminal new model for a layer 5 pyramidal cell in a rat aimed to be multipurpose. Using a detailed anatomically reconstructed neural morphology, the electric properties was fitted to make the model account for a host of electrophysiological data using patch electrodes. Also, new state-of-the-art optimization techniques based on using a multi-objective optimization approach was employed. The resulting model behavior was dynamically very rich, including backpropagating calcium spikes interacting with somatic sodium spikes. The model, which we in our research group call a Hay model, has later been used in numerous labs to study, for example, network behavior, or like in our group, the generation of electric brain signals. Etay Hay is currently leading a research group at the Krembil Center for Neuroinformatics at the Center for Addiction and Mental Health in Toronto, Canada. There he has used the same technique to make similar models for human neurons and explore model networks made of such neurons. In particular, he has explored how such models can be used to understand the biophysical underpinnings of mental disorders such as depression and aid in developing new therapies. In the podcast, we talk about how such multipurpose neural models are built and how he has used them to build network models, explore network behavior, both in healthy brains and in brains of depressed patients. Personally, I found this example of the use of computational neuroscience in clinical applications very inspiring. I think we all have a hope that our field not only can help to understand how the brain works, but also help in finding treatments to alleviate mental health suffering. Note that in the discussion with Etay, I couldn't offhand remember the name of the collaborative project I had in mind for sharing data, analysis techniques, and computational models of neural systems. The name of the project is Open Source Brain, and the link to the website can be found on the episode homepage. If you want to support the podcast, go to patreon.com/theoretical neuroscience podcast and choose a membership of your liking, for example, at the cost of $1 per month. As a member, you will get access to a video version of the podcast recordings, as well as transcripts of each episode. And note that there is a 7-day free trial period when you sign up. I now also have established a YouTube channel called Theoretical Neuroscience Podcast, where you also can watch the videos. I will be most grateful if you become a Patreon supporter to help me fund the production of the podcast, even if you're not too interested in looking at the videos. But I also appreciate all the other listeners, and I hope that all of you can help spread the word about the podcast to other people in the community, on social media or otherwise. Okay, here is Etay. So, welcome to the Theoretical Neuroscience Podcast, Etay.
Thank you for having me.
It's Etay Hay or Hay, or what do you say? I guess you're
Etay
Israeli,
Hay.
right? So,
Yeah.
Etay Hay.
Yeah.
Okay. So, at least the first part of the podcast will be about how to make multi-compartmental neural models. And the reason I invited you for this in particular is that we have been using these multi-compartmental neural models, and I made some for ourselves in our group, but we have really used some of your models a lot. In particular, this model that you made in 2011, at least it was published in 2011 when you were in, I guess maybe you're a PhD student or something working in
Yes.
Idan Segev's lab.
Yeah.
So this was a paper in PLOS computational biology. So there you sort of the goal was to make a model for like a thick tufted layer 5B pyramidal cell from the rodent. So okay, so first, why did you want to make that model?
Well, the main reason actually started by collaborators at the Blue Brain needed better models to simulate. So there was a use case, and you'll see throughout our talk. I think that's a nice thing that people use my models or anyone's models. I think that's very satisfying, and it really gives a motivation. So there was a motivation. The second motivation was that I had questions about neuronal networks, and there wasn't a good model that captured a bunch of the key properties. Until then, models have been used to answer one question usually, and we wanted a bit of a multi-purpose model that integrates constraints. So that was the reason.
Yeah, and why that? Why that particular cell type? It looks really cool, right? I think
Yeah,
it's sort of like a poster boy of of cells, right? So why
yeah.
that particular cell type?
Well, it has been studied a lot, and especially about the two key properties that I was looking for. One was the
Mm,
firing of the cell, and all cells fire action potential, so that was one
mm,
thing, but also the calcium spikes and the back propagating action potential. So because
mhm,
those are thick tufted and also they have thick dendrites, it's easier to record from the dendrites, and there have been more papers that gave us information about the dendrites and their properties. So that was pretty much the best characterized cell, at least experimentally.
mm,
And it's also a cool cell. I mean, it integrates a lot of information
mm.
and it projects an important output from the cortex.
Yeah, and I guess no. I also when you talk about even there are some ideas about how these cells can be used sort of for like in predictive coding and like for
Exactly.
meeting
Yeah.
things at the apical part and the top. Well, what is that about?
So Matthew Larco, maybe you're referring to his paper that
Yes,
he
indeed.
suggested that there can be a coincidence detection using
Mm,
the dendritic and somatic firing. So the dendritic, usually the apical, the distant apical, receives cortical-cortical connections. So they
mm,
can maybe integrate interior or internal concepts and pictures in the brain, whereas the basal dendrites that are nearer to the soma get more of the sensory input bottom up.
mm,
And so when you have both of them firing, the property of inducing a burst firing through the calcium spike and the sodium spike is thought to underlie maybe coincidence detection in the brain.
mm, mm. Yeah. So yeah, and I guess now you also have all these different variations where they explore how the output spike can be some kind of error signal or deviation signal between. But I think there are many candidates or many ideas there, which circles
Yeah,
around this neuron.
yeah.
So okay, so you wanted to make this multi-purpose model that was sort of more, I mean, applicable for many situations, which then was maybe had not been a few of those around before. And then, of course, you need a first thing you need is experimental data, and and I guess you get that from slices, right? Did you do that yourself, or?
No, so we had collaborators, and we also took data from the literature. So the literature is actually very valuable, but
Mhm,
also having a good collaborator that can really you can ping pong about the data, ask
mhm,
questions, get some maybe refinements is also very important. So we got for that project it was from Henry Markram's group. They had a
mhm,
lot of recordings, but we also supplemented it by papers from Matthew Larkham, for example, for the dendritic properties.
mhm. But the recordings, okay, so that was sort of maybe to, but also the patch electrode recordings, the like I guess with one often with one electrode in up in the dendrite and one in the soma. So
Yeah.
they were done mainly in the Henry Markram's lab. Is that so?
Actually, that was more from the literature.
Oh,
The multi, like Henry usually does multi-patch for cell bodies, so he will get
okay.
firing and response from pairs. But
Hmm.
dendritic recordings that has been Jackie
Hmm.
Schiller's group published a lot about the basal dendrites. and Matthew Larkham and a few others have published about the apical dendritic properties. Greg Stewart did some backpropagating action potential and IH current. So we integrated a lot of literature findings, and that's something I'm still doing, and we'll talk about it later. It's not just the literature has a big value, I would say, and people don't appreciate it as much. I mean, they sort of ask questions and look for data that sometimes is already there. It just been, maybe have been published a little earlier in the past. But yes, so from Henry, we got the patterns of firing typical for each cell type, and which is a major constraint as well.
And then, of course, you needed some reconstructed dendritic morphologies. So that's a separate thing. So that you also got from somewhere. Henry again, yeah. So you had these morphologies, and you had like then this different kinds of like essentially membrane potential recordings for different situations of stimulations in the soma or maybe dendritic inputs or something. Okay, so then of course you want to make a model that that sort of reproduces this sort of essentially the behavior you see in experiments. And then I guess first thing, now you have the reconstructed dendritic morphology, and then the next thing is to put in ion channels. And the first choice is sort of what kind of ion channels to include.
So that's an interesting question, because it's a very nostalgical of those steps. It's been a while, but like they're very vivid in my mind. That was one of the first things we did, actually. The first task, asked the Israeli group at the Hebrew University under Idan Segev, and also the collaborators in Switzerland. We set to just really find the best characterizations of the key ion channels in the literature again. So there was a very strong literature curation. Sometimes it was an actual channel with a name, like NAV 1.3, let's say, and sometimes it was more of a current, like delayed, or let's say, persistent sodium or fast potassium. So it's not easy. That wasn't an easy task, and we were sort of aware of the limitation, but we got our hands on about a dozen of the key ion channels. That sort of started the whole initiative that later was published as the Channelpedia, and later Henry acquired a more systematic way to, it's like a robotic measurement apparatus to measure ion channels quickly, but like back then it was the literature mainly. Yeah.
And then of course you need to know then you have to decide on these ion channel types, and you need to know of course assume something about these kinetic constants inside this opening and closing the gates and stuff like that. So that is something you probably take from separate studies.
So the studies give you most of that, and there again, I mean, there are beautiful studies in the literature, people that express a certain protein in oocytes to really better characterize it, or measure currents in living cells. Yeah.
Yeah. And then you need to sort of start, of course, adding, and then you have to assume something about the densities of the various types of channels in the various parts of the neuron. And that's also, of course, that it's also, you also have to make some assumptions there, don't you? That regarding that, you cannot just let all densities vary freely in the optimization procedure, right? That's
right.
probably.
So we can maybe talk a little about the optimization, which is a big step there.
Yeah.
You do indeed need to provide some search boundaries. They can be, they don't need to be too narrow, but there is an importance not to get them too unphysiological. So we got the ranges, but then we used a very important tool that I've been using since, and that's a genetic algorithm. And it really worked well. I mean, the solution space, or the problem space, if you want, of what densities will give you the right firing behavior and electrical properties of the cell is pretty complex. And genetic algorithm does a really good search through it. and it's automated. So we've been able to build the framework, and then it can be reproduced more easily to find the densities that give you the behavior.
Yeah, I guess this there's something called the was it maybe older? You mentioned it, the blue pie opt. Is that something that came
Yeah,
out? That is
it came
widely
out
found, right?
later.
Yeah.
So
Yeah.
my work was the beginning of it, and then
Mhm,
they made a more of a Python package and a tool,
mhm,
because the blue brain, then I optimized the key, maybe one of the key neurons that we were interested at, but then
mhm,
they replicated the approach to model a lot of other cell types. And
mm,
because it's an automated approach, it's a bit more easily transferred instead of manually fitting and
mm,
refitting every cell type and doing it many, many times just manually. The automated part was very important. Yeah.
mm.
So that's BluePyOpt is a good tool. We use it now, and it's free.
Yeah. So, but then of course, then you have to decide on a cost function or error function. What does a good fit mean? You need to quantify, make some choice about quantification of the error, and
Yeah.
then sort of the naive thing is maybe look at some kind of square deviation of the predicted membrane potential, for instance, compared to the measured or something like that. But that is not such a good idea. So
Yeah.
why is that?
Well, I think it's good maybe for some things, but all in all, first of all, the cell is not repeating exactly the same voltages every time you give it exactly the same. I mean, you can give it the same current 100 times, and it's not going to do the same thing every time.
Yeah.
So, a bit more importantly, we extracted features
Ah.
like firing rates, adaptation index, spike height,
Mhm.
and nicely, part of that framework is you extract the statistics, the mean, the standard deviation. So
Yeah.
your evaluation is very rigorous using those
Mm.
statistics of the goodness of it.
Yeah, because then I mean, so for each, then of course you have like, I guess you have many features. So in some sense, you have this, you optimize some kind of sum of errors over all these features. But each of these are weighted by the inherent experimental variation. Yeah. If there's some kind of feature that varies a lot in the, like for a single neuron when repeating the experiments, it doesn't really make sense to fit it to one of these, like just one observation, right?
That's true. I mean, and also that way you get a model that you can put within the context of a population. You can say,
Mm.
this is within the range of what I see in pyramidal neurons. You're
Yeah.
not trying to fit an exact single cell. Sometimes there is a motivation to do it, but many times you just want a typical or a pyramidal or a pyramidal that is physiological.
Mm.
I'll emphasize also that another tool in conjunction with the genetic algorithm was multi-objective optimization, where
Yeah.
each feature is, we don't do a weighted sum, which has a lot of disadvantages. We
Okay.
can fit all the features parallel in a way that they don't interfere with each other in terms of, and we don't need to come up with like a weighting for them. And then, as you said, if there is a feature that varies a lot, and then it just, its standard deviation will be just affecting its own fitting, and it won't interfere with the other one. So
Yeah.
I want to also take the opportunity. This was work with other people with it from Idan's lab. Shaul Druckmann is one of
Mm.
them.
Yeah.
So I developed a lot on my own, especially the dendritic things, but we were
Mm.
working as a team about it
Yeah.
on that.
And then, okay, so then you had like this multi-objective optimization and all these different sort of like error measures for like, I guess about 20 different targets or something, and some of them were for sort of responses to when you stimulate in the soma, right? Like step currents into the soma, and different step amplitudes, I guess, and somewhere for rather for stimulating another behavior of the neuron, meaning that some and then input to the dendrites. Is that so?
Yeah,
Yeah.
yeah. So we fitted the backpropagating action potential. So if we induce a spike at the soma, we constrain the neurons to have the right attenuation along the dendrites. which is not passive. It's a bit more moderate attenuation due to some active properties of the dendrites. And in addition to this, we model this coincident burst
Mm-hmm.
behavior, the backfiring that was characterized by Larcom,
Mm-hmm.
by stimulating both the dendrites and the soma within some kind of delta T.
Yeah. And then I saw that you have altogether 22 fitting parameters. Uh,
Awesome.
yeah, well, I read your paper
You
today.
counted? Yeah.
No, yeah. So no, I read your paper today. That's what I usually
Yeah,
do before
that's
interviewing
good.
people.
Yeah.
So that's good. So yeah, 22 parameters, which are essentially free. But I mean, as you said, you have some. I guess sometimes I saw that you put some lower and upper limits on these parameters, describing all these. Well, I guess channel densities and some time constants and so on. So I guess
Hmm.
that was taken from the literature or some other constraints.
Yeah. The limits were, I mean, there was some knowledge about what is the density can't be so high, right? I mean, there is a limit to how many channels you can pack in one unit area.
Mm.
And the lower limit also, I mean, either it can be zero or you can force a certain kind of minimum as well. So there was, I would say it was informed. There aren't really hard constraints about it, but it was
Mm.
informed.
Yeah, cool. So then of course then you have these 22 parameters and I remember we were involved in some kind of fitting of pyramidal cells, actually with Tumo and making Martin in our group, and that was like just a few years ago, much after your work. And then I remember it was sort of like important to have some kind of that we first fitted sort of like some like the passive parameter, like a passive membrane resistance, actual resistance, and capacitance, and had that first, and then that is some kind of sequence in the fitting of parameters were important.
Yeah.
It was important. Is that also something you had, or?
Yeah, so and I described it in the paper that we did a two-step fitting. One was for the calcium firing, if I'm not mistaken, and then the
Mm,
somatic firing. I don't think it was the other way around.
mm,
Um, it just that the solution space is more complex, and you will need a lot more iterations to try to really traverse it completely. So you sort of want to just constrain it. In another paper, I also fitted really the dendrites, like the passive properties first, like even sometimes manually you can quickly do it. You fix the parameters. So yeah, definitely it helped. Uh,
mm,
if you have mild constraints for the dendrites, you can do it in one go though, and we have done it.
mm,
But for the calcium spikes, it's not really the case.
mm. Yeah. So then you had like this. I saw that like this. You did this fitting in steps, and then you ended up with some kind of well model that you were happy with. This as a first go, and then I guess one thing that you would like to be. I mean, as you said, the goal was sort of to make some kind of multi-purpose model that was sort of applicable to many situations. Not maybe not only the ones you have fitted it to, even though they sort of covered quite a bit of ground
Hmm.
with inputs all over the place. So
Hmm.
you also try to test it on sort of like a new some kind of new target behavior.
Yeah. So in that same paper, there was a nice validation that when you stimulate the soma with a train of pulses,
Mm,
beyond a certain frequency, that somatic train can lead to summated EPSPs in the apical dendrites that trigger a calcium spike. And this
okay.
has been another nice work by Matthew Larcom, I think. And the model wasn't constrained with that. There was a pure validation, and it
Mm,
bang on reproduced it. So yeah,
cool. Yeah.
yeah.
So so you are sort of well. So yeah. So you how 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 multi-purpose do you think it is? How much? What can
It's a
we?
good question.
Yeah. It's
Yeah, I
because
think
you
it
were.
probably has its limitations. I think there's some people that concentrate on one very, let's say, molecular aspect, maybe the calcium buffering or something that is very particular. and therefore may need a little more refined models on that, right? But I think in terms of physiology and firing and mostly the electrical properties, I think it has behaved well. I don't think it's a done deal, definitely, but has been used by a lot of people and was useful for them. So that shows that it served well. But I think I would definitely see a point in maybe trying to introduce a few other things to it and maybe even test it a bit more.
Okay.
Yeah.
New experiments.
So
Yeah.
yeah, like I mean, how does it do with ramp current? How does it do with IH resonance, right? Behavior.
Because IH was in the model,
It
right?
was, and for the most part, and by the way, there have been groups that just used it to test, and they've reproduced their properties that they recorded using our cells. And again, this wasn't constrained for. So in many cases, it is multi-purpose and it's holding.
Yeah. We have used it a lot, but to compute sort of like local field potential from the IH. But of course, that's an application that it's not so easy to validate it based on that. We just sort of have to we have to sort of take it as is and sort of see what it predicts. But one important thing here is that when you do this multi-objective optimization, you don't only get one parameter set that sort of like is the clear winner, right? You get a bunch of parameter sets that are equally good, or do equally good predictions, right?
Yeah.
And but that's probably that's maybe like a feature, not a bug.
Exactly. And people have shown it. If Marder showed it with like the cells from crustaceans, where cells that are even functionally very specifically conserved, like it's a specific cell of a specific circuit, they could do the same behavior with different combinations of densities. And not to mention, as we said, that pyramidal neurons are not all the same. They have a range of properties that are more similar to each other than other cell types. But there might be a pyramidal that fires a little faster, one that fires a little slower. And the nice thing is that the genetic algorithm gives you a population of cells that you can then take advantage of and even ask questions about.
But this population, I mean, they have the same overall error function, right? So in principle, they should spike similarly also, shouldn't they? I mean, they should. So are
The
they really different in their
successful
behavior? Yeah.
They do. They are. The successful models, as I think I call them in that paper, are ones that are physiological. So, as I said, like when you record real neurons, like pyramidals, you will get a range of firing rates in response to the same current. You have different wheelbase and different properties, and the population of models that are successful are not the same necessarily. They fall within that experimental range. Now, some of them are similar. Some of them may fire exactly the same output per given input, but not necessarily, which is great. Like you essentially get in many ways, the experimental variance in your model as well.
Yeah, maybe it also reflects that. I mean, that it has all this. If say a neuron like your neuron is supposed to have a particular function in terms of how it integrates inputs and produces spikes, if there are many combination of parameters that fixes this, it makes or are able to do this or does this, then it makes also the cell more robust to damage, right? I mean, if something bad happens to
That's true.
one ion channel, if you have some kind of homeostasis mechanism. So I think if it was like, and I think that's often I've heard that critique of this sort of like this multi-compartment model. There's so many parameters, but that's because I think that from physics you're more used to sort of like simpler models where like the parameter set that fits whatever best is sort of unique, and if
Exactly.
it's not, something is wrong. But I think that's just different in biology, I think.
Yeah, I agree. Yeah.
Uh,
Biology is noisy, and usually it's a strength of the brain. It can not only that it's robust to perturbations, so it
mm,
means if you just change the conductance a bit, things wouldn't fall apart, especially if the other conductances compensate for it. But by having a far like a population of neurons that behave a bit differently, it
mhm.
allows them to compute a bit differently, and the whole firing and circuit dynamics is becoming more versatile instead of being epileptic. Like if all the cells
Yeah.
are clones of each other, it actually, some collaborators of mine shown that it makes the network less robust.
Huh. Hmm. So yeah, so and I think I mean so one goal of this is obviously to be able to have to make this multi-purpose neural models that you can use, for example, to hook up in a network. But another thing is to actually get. I mean, you can sort of look at the parameters of the the resulting fitted parameters, and maybe learn something about because that's a different thing. In principle, you could have a model that predicts the spikes well, but nevertheless is not so faithful when it comes to predicting that the parameters doesn't really correspond to the underlying biology, right?
Exactly. Yeah. We
Mm,
were able to show that, for example, the range of apical sodium
mm,
densities in the models
mm.
that is important for backpropagating action potentials agrees with the estimates that people have done experimentally. And those
Okay.
estimates are not easy to do. And that's exactly what you say. I mean, we can use the population of models to give estimates of the ranges of densities. And as you also said, that those ranges are physiological. It's not, and that makes them a little bit more maybe capable of generalizing, hopefully, right? I mean,
Mm.
if you really have a non-physiological value, it might do something wrong down the road, right? So with some
Yeah,
kinds of
that's true.
behavior.
At least if
Yeah.
there's one parameter that really looks strange, like
Hmm.
the capacitance, which is 10 times larger than what you're used to,
Exactly. Yeah.
then you start something might come back to haunt you if you use
Exactly.
it for something new. You also tried to use the model fitted to one morphology to other morphologies, right?
Yeah.
How
That
did
was a
that go?
follow-up paper.
Yeah.
I like
Yeah.
that paper actually a lot because it was a known problem. I mean, you make
Mhm,
a nice model, and if people want to simulate a variety of morphologies, it
mhm,
fails often, more often
mhm.
than not. And we found a simple rule based on the drain of the current into the dendrites compared to the somatic properties, and you can scale your model to make it work on a bunch of morphologies. So, yeah.
Ah. So what paper was this? Because you briefly mentioned it in this 2011 paper. There was like a later paper.
Yeah, it's a later paper. I think it's neurophysiology from 2013.
Okay.
Yeah,
Yeah, I'll link to that
yeah,
also on the
it's
website of the
yeah,
episode.
yeah,
Yeah.
yeah, and it has been used by a few groups, including the Blue Brain, because they have a lot of morphologies, but not necessarily they want to fit models every time to a different morphology, and that's a good way to scale it.
Yeah.
Yeah, it's
Exactly. So how because now you have. I mean, I remember when I when I grew up. That's maybe a little bit, but when I sort of started doing modeling in the 90s of neurons, you had a handful of cells like Men and Sanofsky and had like few
mm-hmm,
cells and they were available, and then you yourself, for example, came along. There were also other layer 5 cells around, but
mm-hmm,
then it was only like a single or a handful of neurons. But now you get, if you go out to databases, I guess like the Blue Brain or the Human Brain Database has a lot of fitted neuron models, and also the Allen Institute
mm-hmm,
has a lot of fitted neuron models. How and that's essentially made automatically, right? Some kind of
mm-hmm,
automatic procedure. Have you
mm-hmm.
how how robust are these automatic procedures? I mean, I'm not quite sure if you have explored this, but because you had a quite a bit, I mean, manual control and component to study whether it made sense or not.
Yeah, it's a good question. I think they have value and they reproduce things, and sometimes it's necessary. I
Mm.
do think that still the manual curation is important.
Yeah.
And I think that, especially the two-step model that has calcium spikes, that has not been really replicated in any of those databases. So there
Oh,
are some tougher beasts of neurons and
yeah.
properties out there.
Yeah.
But like even as we may talk later about the human models,
Mm.
the Allen brain has fitted using the same tools from the blue brain, the blue PyOpt, and the approach. And we were actually, when they just set up their framework, we had good conversations to essentially, you know, discuss how we did it, and then they took the approach as well and made improved it the way that they wanted. But those models are good as a like batch, giving you a lot of models, but it's very there is still value to that careful, personal, I would say, calibration of
Yeah.
the automated process, and that has always been the case. But in many times, once you have the right calibration figured out, you can really reproduce things, and
Mm.
so it's a balance between the what the automated tool gives you and what still needs to be curated as a scientist,
Mm.
right? A human intervention.
Mm. Because I guess most of these like automated pipelines, they only have uh, current injection into the soma, don't they? Or
Not
do they also?
necessarily, but most of them stick to that. That sort of maybe speaks to some of the general issues. Is that sometimes people just make the simple models, let's say
Yeah.
that reproduce the somatic firing. A lot of the models out there that have been made like by like a lot of models at once and
Mm.
don't necessarily capture some properties.
Yeah. Mm.
But I think as a community, with by discussing and collaborating, and if there is if the users are interested, then essentially you can really tune your automated tool to demand a little more than the models you produce. Yeah.
Cool. So now we're going to more like this was has so far been about making these models, of course, which I'm yeah, very appreciative of. We have used your models actually in our book, Electric Brain Signals. We have like the Hay model is used throughout as an
Mm.
example model. So we are very appreciative for that. But then there was also this more recent work, I guess, in your group. Now you're now in Canada, aren't you? Where are you in Canada?
Yeah. In Toronto,
Yeah,
at KMH,
yeah. CAMH. That's can
Center
mental.
for Addiction and Mental Health. It's
Okay.
actually a mental health hospital that has a strong research component as well.
Okay. So it's not a it's a part of university or is it
Yeah,
separate?
it's
Yeah.
fully affiliated with the University of Toronto.
Okay. Okay. So now you work now on mental health issue, and then so this was actually something I just recently learned about actually from talking to you that actually there are sort of like this you have studied like putative role of some of these interneurons. Well, there has been some neurons which are whose activity has been associated with depression, and so then we suddenly have like a handle to make a link between models of neurons and neural circuits and sort of yeah and like this obviously this psychiatric disorder. So tell me about it. What is so there is this role of SST interneurons.
Right. So when I started my lab, there has been a nice opportunity that there is some data about circuit mechanisms of depression and other
Mhm.
disorders, but there haven't been models. There was also an opportunity that there is a lot of human data, neuronal properties, synaptic properties from tissues, human tissues that are used for electrophysiology that haven't been a lot, I would say, modeled. I mean, the Allen brain started making a nice library of it, but there was much more to do still.
So
So,
so
yeah,
these are tissue that has been sort of taken from like patients.
Yeah.
Because as part of some other clinical
Yeah.
treatment, right? Medical treatment. Okay, and then you
Yeah.
study the main the slices. Mm.
Yeah, so if there is a tumor or a epileptic focus somewhere deep in the brain, you go through the cortex that is mostly healthy. Obviously, that's not 100%, but for it behaves actually like healthy cortex,
Mm.
and you can use that extracted tissue to do electrophysiology and get some properties about the human microcircuits.
Yeah.
So what we've done in my lab is to two things. One is generate those detailed models of human neurons now and human microcircuits,
Mm.
and that means the properties like the L5 pyramidal from the rodent using similar approach, but also circuit properties. So connection probabilities, cell type proportions. And
Mm.
there are a few key cell types. One of them is the SST interneuron that you mentioned.
Mm.
And collaborators from the same institute have actually, in depression patients post-mortem, check the expression of SST in those interneurons and saw that
Mm.
it's reduced, linking
Okay, so what they
essentially.
do then, they
Yeah.
look at the expression of these, the number of these. No, about the number of these neurons. No, no,
So
the
interesting.
yeah. Go ahead.
Yeah. Interestingly, the number is not changed. It's
Mm.
more that the expression of SST that is linked to essentially a weakening of the inhibition.
Ah.
That's what they found.
So the S is first now you have okay. So you have essentially one type of pyramidal cell, or that's sort of like to make it simple. But they have three main types of interneurons, right? That's that
Yes.
PV
PV.
and SST and I guess VIP, or it's called something else.
VIP.
Yeah.
Yes.
So this now we're talking about one of these three like main subtypes of interneurons, the SST interneurons, which has been
Yeah.
then as associated with depression.
Yeah, and interestingly, SST interneurons pretty much mainly target the apical dendrites, the distal part, which we've been talking about in terms of calcium spikes. So there
Mm.
is a lot of interest in, and they're modulating an important part of the pyramidal neuron integration apparatus, right? So I would just quickly say that we haven't exhausted all the questions about it, especially
Mm.
because there is a lot of interesting things to check about
Mm.
this reduced inhibition. But
Mm.
we did two important works. One of them about more like the firing in the circuit that is affected by the reduced SST inhibition. And
Mm.
one is actually a little bit more recently. Now we have a new paper coming out at iScience
Mm.
that looked at dendritic integration, where we constrained the models, a new generation of those models, to reproduce BAP as backpropagating action potentials as measured
Mm.
in humans.
Yeah.
And then we check the effect of SST on that.
Yeah.
So yeah, I'll emphasize that there has been theory about what happens in depression due to that dysfunction of SST, but it hasn't been tested in humans. It
Mm.
has been indicated by some rodent experiments.
Mm.
But yeah, so our study had the application to test this hypothesis in humans and then
Exactly.
do
So the
something further.
yeah. So exactly. So the okay. So the goal then, if sort of well, depression is associated with too little inhibition coming from these SST interneurons, you can imagine some kind of therapy down the line of doing something to increase that inhibition
Yeah.
from like to increase it, or yeah, something like that, right? So
Yes, and that has been, I would say, two important themes in my lab following up that have been to, first of all, find some biomarkers in clinically relevant signals, such as EEG, and that's where the LFPI tool has been very valuable for
of
us.
course. Excellent,
It's
very
nice, I mean, in this conversation, you see
uh,
you've used my models, I'm
uh,
using your tools. I mean, I
uh,
think it's very satisfying
it is true.
right?
And it also
Yeah,
feels good to when you do put in all this work in a thing that other people actually take
yeah,
advantage of it. It's
yeah,
fantastic, right? Yeah. Yeah.
yeah.
Mm,
Otherwise,
it-
it's a
mm.
tree that falls in the middle of the woods, right? And nobody hears
Yeah.
it, right?
No,
You can do
exactly.
an amazing work, but
Yeah.
then
Yeah.
So that was one thing, and we are actually testing those. We do show that even though it's a microcircuit,
Mm.
right? It's a microcircuit level change. It's not like the whole connection from the two hemispheres has changed. There
Mm.
is a specific interneuron inhibition. It affects the whole circuit, and
Mm.
that gives you detectable signatures in EEG.
Mm.
And they can be used to stratify patients and diagnose. And here I will emphasize not every depression patient will have reduced SST. This is one mechanism.
Mm.
Depression, like many other mental disorders,
Mm.
is complex. There is different types.
Mm.
Some people, it's a serotonin thing, and then the famous, SSRI medications can help them. But 66 or so percent of the patients don't improve using the existing methods, which leads
Mm.
us to the second thing you alluded to is the new treatments. And the
Mm.
same collaborators that have been on some of my papers from KMH have developed a drug that boosts this very specific inhibition
Yeah.
by targeting the receptors on the apical dendrites that it links to, and they show that it's good. It's very promising in rodent studies.
Mm.
It really they recover from a lot of their cognitive symptoms and mood
Mm.
symptoms.
So how this is sort of I'm not so familiar with all this like the clinical work, which is of course one of the main motivations why we work on this. This
Hmm.
I great if you can help this huge patient group and society as a whole. So, but these models for like the depression in mice, how well are they established? I never. This is always, I guess, a challenge, right? You have this.
It's a good question. You
Yeah.
mean that the mice is a model of human depression?
Yeah.
It's a good question. Frankly, I didn't get as much into it because my lab and approach focuses on human models to
Yeah.
study human disorders, and that's exactly the motivation, you
Yeah.
know?
Hmm.
And I would say that they do, there are equivalent symptoms that you see in depression. And so they do replicate a lot of those symptoms. So
Hmm.
normally you wouldn't necessarily call it as a model of depression, but
Hmm.
more of an animal model that replicates some of the key symptoms that you see.
Okay. Hmm.
And there are similarities. That's part of what we saw along the way is how similar you have the same cell types. You have a lot of properties in the mammals are conserved to a degree, right? But SST is actually, as you know, we have data from human patients that SST is a potential mechanism, and
Hmm.
if you affect SST in rodents, you induce depressive symptoms. So yeah, there
Ah.
is a nice growing body of evidence.
Hmm. Because I thought that in order to do this model study, I mean, you made new human cells. You made like one for a. This is a paper from 2022, I think, with the Yao
Yes.
as the
Yeah.
first Cell Reports. Yao was the first
Yes.
author. So you had made one model for pyramidal neuron, which didn't have back propagating action potentials, as I understand it. Like that version,
You're
maybe
right.
that's like a new version.
Yeah.
Yeah,
So the new version at iScience has, but this one didn't. You're
no.
right.
And then you also made models for the this like the SST interneuron and the PV interneuron and the VIP interneuron, right?
Yeah.
And that was like a similar approach as for the 2011 paper, or
Yeah, to some extent, and that's the nice thing. I think that's the whole point of automating. What you do is that you don't need to redo a lot of the things. You don't need to manually fiddle, but there has had to be a little bit of thought going
mm,
into it. There are some assumptions that you need to make. You can't just dump complete freedom on your genetic algorithm.
mm,
So we made a few assumptions. For example, do you model a little bit of the axon and things like this, right?
uh,
But for the most part, it is a conserved approach. I will very importantly thank the Allen brain for sharing a lot of the human data, a lot of those interneuron properties. We pretty much took the experimental data and refitted. Yeah.
yeah, Alan Institute is really cool.
Yeah.
So
And
that
we take the same approach, by the way. I mean, we try to share our tools as much as possible.
yeah, exactly. So then you had this. So you were able to make these different then human neurons, and then and how. I mean, it's also interesting you said this thing about you can sort of almost start a little with the rodent model and then
Hmm.
sort of tweak it to make it more human. Of course, the
Hmm.
morphology is a bit different and so on.
Hmm.
And it's almost like this foundation model they use in the AI, right? They
Hmm.
train
Hmm.
it and then they have like this foundation model and they don't need, and then they can sort of tune it to different applications with
Yeah.
a little bit of training on top. So maybe it's well, maybe it's just an analogy at least.
Yeah,
So
I will emphasize that we did fit it from scratch, but use the same approach, right?
okay,
Although, yeah, so it's
mm,
not a modified model, it's just from scratch.
okay,
Also, let's remember in human, most of the data is available from L to 3,
yeah,
layer to 3. Although we did already published an L5 human microcircuit models with all those key cell types. That's in 2023. It's cerebral cortex, studying aging as well. But yeah, so it's the approach, it's the whole like
I see.
pipeline where you just put the features, you get the model.
Okay. Because I mean, that's the layer 2, 3 cells in humans. They are really big and have big dendritic trees, like where the layer
Exactly.
5 don't have this
Yeah.
big. So
Yeah.
they have like a little bit. They look a little bit more like in layer 5 looks a little bit like an old Christmas tree, but lost all the stuff at the top.
Exactly. I was really like surprised, and I was for the beginning year of getting into human neuron modeling was really
Mm,
trying to get my handle on it. Is it just
yeah,
because we don't have data? Is it because it has been cut? But it is actually the case that most of the L5 are pretty sparse. There are
mhm,
just a few, a small percent of thick tufted ones,
yeah,
whereas the L23 are pretty heavily tufted, which maybe
yeah.
shows something about the circuit. It's an
Something
open question.
slightly
Yeah.
different. Yeah.
Yeah.
Because so they are so just looking at the dendrites and the different layers, you see a difference between human cells and rodent cells. But what about the like the electrical properties? I remember there was something about that the capacitance of human cells could maybe be smaller or something than like half of rodent cells, or not quite sure how
Yeah,
that has sort of.
it's a bit debatable, this thing.
Mm,
I think we adopted the same value for capacitance because there has been enough groups that supported that. So I don't think it's clear enough that. But
mm,
definitely in L5, for example, the just electrically, the
mm,
soma and the dendrites, the distal parts are more decoupled, just owing to the length. And by the way, that's where the that group forgot the name, but like they published a paper that showed it experimentally and then used my rodent model, stretched it, and showed that it can sort of replicate the human
ah,
behaviors.
yeah.
So
Oh. Hmm.
yeah, so
But then you in your model, then you wanted to make like a circuit where
yeah,
the prime the star neuron was this wonderful layer 2, 3 pyramidals, and then you
yeah,
had these 3 types of interneurons around it, and I guess you had like
yeah,
1,000 neurons about, and then but then you have to make some assumption about the connections, right? That's
yeah,
which you didn't have to do in 2011 when you only considered a neuron.
exactly.
Yeah.
And I'll briefly mention, and you have used my circuit model from the rat as well, that was sort of simple and more not as thoroughly constrained, but this
Okay.
time we went through the literature, and there is a nice human literature from groups like Gabor Tamas that recorded interneurons and properties of pairs. So people not only just get human single neuron data, but pair
Mhm.
properties, which is pretty much the golden standard for synaptic properties. And we integrated all of this. So we have data for SST to pyramidal, PV to pyramidal, and pyramidal to pyramidal that already covers a lot. That's
Mhm.
really from human.
Yeah.
Yeah, so integrated it all. It was a fun thing to do, which I haven't done before. Like before, it was like make the single neuron model,
Yeah.
hook like 150 of them with some kind of probability, and let's ask questions. But here, yeah, we did a good constraint for the synaptic probability
Mhm.
and connection probabilities we know from human, pyramidal to pyramidal, but unfortunately, still most of the rest of the data for that comes from rodent data.
Yeah.
Yeah.
Mm.
Although recently there is improvements in getting higher yield of a lot of pairs
Mm.
from human data, but it's still in development.
Mm. Yeah. Do you have any systematic way of? I'm just thinking about there's what is this website in English? What's called the brain net? No, there's some kind of network where you can sort of have a model that sort of develops and sort of is when new data becomes available, right? It's not that the model doesn't sort of stop developing just because
Yeah,
paper is published.
it's a good question. By the way, I think that to some extent, mainly the blue brain, maybe also the Allen brain to some extent, take that approach where they sort
Mm.
of keep developing their model, and there is definitely a motivation for that. But for us, it was more like it's a little bit more frozen for us. We get a decent model that we're happy with, and then maybe 2 years, 3 years down the road, we have a big change and we make a new model. So
Yeah.
it's not so much of an iterative process for us.
Mm.
I think that, unfortunately, right, because like it means that most of the key properties we wanted are there. But I think, as like I said, we just 2 years later or so came up with models that have dendritic properties of human pyramidals. So that's a
Mm.
new version, right? And
Mm.
when we know more about human properties of synapses, like connection probabilities, and there'll be the third, maybe, generation of the human model. So
Mm. Yeah.
I would say it goes by leaps
Yeah.
like that for us.
Yeah. No, but of course, it's also as a question of resources, right? If you
Yeah,
have
exactly.
some kind of setup for continually upgrading the model,
Exactly. Yeah.
that requires resources also. So,
Yeah.
but then when I go back to the 22 version, then so you hooked up, made this new network, and then uh, you would, I guess, the idea was to look at differences between the like the circuit behavior, network behavior. I think you had around 1,000 neurons in like whatever, like a baseline healthy control kind of situation and the depression situation. So what were sort of like the key differences or key features that you look for?
Yeah, so like I said, there was a hypothesis by people that, studied in rodents or things like that. And the main hypothesis was that because SST interneurons are very important in baseline firing as opposed to response, a reduced inhibition will increase the noise in the circuit. So, and
Mhm.
then the signal to noise ratio will decrease. And that's exactly what we showed. We obviously were able to do it systematically. We can reduce the inhibition to different degrees and show the relationship. So I would say it was a systematic demonstration or
Mhm.
mechanistic linking between the reduced inhibition and circuit effects, like
Mhm.
for activity that hasn't been done before. Even more importantly, we translated that change in response and baseline to things you can measure in the clinic, like signal detection. So if
Okay.
you,
So what
yeah,
is the thought there that you, if it's more noisy, then if you get a weak sensory stimuli, you won't notice it as
exactly.
well, and that is also a feature when you are depressed or for depressed
Yeah,
people.
yeah. So
Yeah.
depression patients tend to think a bit more inside their head. They're a little less attentive, they're a little less, and some of the hypothesis is that their cortex is a little bit more noisy. And
Ah.
therefore, when there is a little weak signal received, the firing is not so different than that noisy baseline. So another brain region that has to read out and say, okay, there has been a signal, that circuit reported it,
Mhm.
is not able to do it so well. So we were able to show that
Mhm.
the effect of reduced SST can explain the twofold or threefold increase in errors you see in patients.
Yeah. So that's what a failed signal detection and false signal.
Exactly.
What's the differences there? Failed is when you don't observe a stimulus or don't notice a stimulus, maybe.
Yeah. So there was a stimulus and you didn't notice it. And
Mhm.
false is that there isn't a stimulus, but you think you saw something. And
Ah,
I won't go far into the
no, no, it's
psychiatric
like the
terms, but it's more
psychophysics.
like an illusion, right? It's a
Yeah,
bit of an illusion and
yeah.
also, yeah.
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 So, okay, I think this is really exciting. This is also like an eye opener for me that you can
Yeah.
actually use this kind of models, um, to well, this kind of circuit models to study, even maybe get a handle on some of these like psychiatric disorders and suggest treatments. So you
Hmm.
also looked. You also mentioned that you have studied other aspects like aging.
Yes.
So what was that study?
So there are, I would say, two studies. The key one that has been published already, we looked at human neuronal data of the type that the Alan has, that our collaborators in Toronto have. We're working in collaboration with neurosurgeons also to get data directly from them. And we analyzed the IH current, the SAG current, that is an
Mm,
indicator of the IH in the dendrites, and we saw a change between younger humans and older humans. So, and the nice thing, every patient, or I would say every human data that you have, is has an age parameter that is associated with it. So
mm,
there are not a lot of things maybe you can ask about human data, but aging is actually one of them. So
mm.
that's pretty neat.
But are there any so like this? Now we talk about IH, which has nothing to do with physical age. It's a different. It's like
Yeah,
the capital H or the letter
exactly.
H. Yeah.
Yeah.
So what? But is there any particular link between IH and something that like some kind of
Yeah, yeah.
loss
I'll
of
give a bit more details. So what we saw, and that agrees with what you see in monkey study,
mm,
is that the IH is increased, which actually dampens summation of PSPs,
yeah.
and therefore the activity in the cortex at baseline is reduced. IH is mostly active at baseline, low
Mm,
firing rate and sort of like potentials, and that agrees with older monkeys have lower firing compared to younger monkeys. So that was a nice link
okay.
again between one ion channel in the dendrites and things
Mm,
that you can see in the network.
okay. So but it's not directly related to the mental capabilities of age.
The links remain to be established. I
I see.
think that that was beginning, and that's maybe where I can mention our next work because one of this has been done one of my most gifted students, my postdoc Alex, and I told him I really want to find a way to link a cellular mechanism or circuit mechanism to a functional deficit, right? Make a bit more interesting links because
Um,
the dampening of baseline firing, we had a hard time seeing the implication or suggesting the implication of that. So we did in a study that right now we're drafting for publication. Uh, we did a literature survey of all the mechanisms that in human studies seem to be changing. We came up with a bunch of them, including NMDA, synapses law, some of the very classic ones, and we now are more able to show the effects on function and also EEG. In
mm,
the same sort of approach that we took for depression, there are EEG biomarkers, and they actually replicate some of the EEG studies you see in older people, which
mm.
which is pretty neat.
Yeah. Cool. So yeah, now that's and you also I think you had started to do something with towards another well a and like schizophrenia, that's a
Yes,
psychiatric illness.
yes.
What is the approach there, or what is
So
the
interestingly, in schizophrenia, it's the different interneuron type, the PV, that is implicated,
mm,
and there are a couple of mechanisms involved. Again, it's all human. My approach, at least, of is let's go find human indications, right, in human mechanisms. And there are nice studies, post-mortem studies, that really give you the candidate mechanism, and it's really a low-hanging fruit to model and link to things.
mm,
So we were able to, and this is on preprint, and soon we'll submit a revised version of it for peer review. But it's a
mm,
very nice work that link the reduced inhibition to the main EEG biomarkers that is
mm,
actually used by a lot of groups, the MMN, the mismatch negativity amplitude. And obviously, again, like in depression before, people have hypothesized, that PV is probably playing a role, but there hasn't been a clear mechanistic demonstration using detailed simulations. Obviously, currently we are limited in doing that experimentally. That's exactly where those models have a utility, and we were able to show that.
Yeah. Very exciting. I mean, one thing, and now I'm back a little more to the general thing of modeling these I mean, neural networks, right? Starting with biophysically detailed neurons and everything. And of course, there's when you make a neuron model, there's sort of like uncertainty and some kind of always some kind of errors involved, right? And and when you hook them up, and there's also a new set into neural networks. There's a new set of worries and errors and stuff. And often when you sort of say, for example, the Allen model for visual cortex, Allen V1 model, then they keep the neurons fixed and they typically vary the synaptic connections when they fit into the network behavior. And that's of course also what they do in AI, where they keep these ReLU neurons fixed and just change the synapses. Is this a healthy approach? Is it doesn't it maybe doesn't really matter so much if you fit one thing or the other because
I would say, first of all, this has been our approach as well.
yeah.
We have models for single neurons and also synaptic properties, and then we can change circuit properties like
Yeah.
background activation of the different populations, a little bit of the probabilities of connections, and maybe the weights.
Mm,
So actually, it has been our approach as well. I don't think there is a right or wrong, or I think, I mean, if there is a reason to change the model, then probably it makes sense. The thing is, again, it's the same thing as fitting calcium spikes and sodium spikes at the same time. If you had to change the neurons and change the connections, I think it's a little too much for
mm,
the current tools and maybe even our current grasp of the fitting and performance and all of that. I don't think it's a wrong approach, but I personally am more of a circuit person. As much as I value the properties of single neurons, I
mm,
always think about them in the context of how do they affect the network, right?
mm. Yeah.
And ultimately, our networks are super important. So I'm
Mm,
sort of thinking at the level of scaling the weights, and
yeah.
I will mention one more thing that might be a topic for a further discussion. But one of the key things we're looking to get into is being able to actually train our detailed models the same way you train more simple neuronal network, right, with TensorFlow and all of that, but to train them to do functions. So instead of
Mm,
using a more manual approach, be able to teach them to have the weights change. And
mm.
in which case, I think I would be happy to keep the neurons the same because otherwise it's going to be hard.
That's
It's
true.
already hard as it is.
Yeah.
Yeah.
No, I think there was. I mean, there was this. I think exciting paper by this name was first author was Chen. It was in the group of Wolfgang Maas, where they actually
Hmm.
took this old
Yeah.
LNV1 model and turned it into a TensorFlow model, so they could train it to
Yeah.
do
Yeah.
functions. Yeah.
Exactly.
So I think
Yeah.
I agree. That is sort of an exciting thing. But I guess also when it comes to like this two stage with calcium spikes versus sodium spikes. I mean, it's also like different time scales, right? So maybe they don't connect so directly, and maybe it's similar thing with neuron dynamics and network dynamics met some kind of I don't really know, but this is something, and I guess if you have limited data to fit a model too, you can probably tune if you have a starting point and tune the neurons or tune the network, maybe you can be quite successful regardless.
Yeah.
Yeah, maybe. So
And
that's
if you
also
think
the
physiologically, maybe, right? I mean, the neuron may not change so quickly every millisecond, right? So there is probably a motivation for this. But obviously, there is plasticity. I mean, our models have short term plasticity, which is the mechanism that is relevant at those very quick scales, right?
oh. Oh, so you have
Uh,
short-term plasticity in your network models?
the synapses
Yeah.
facilitate or
Yeah, yeah.
depress,
Very
which
good.
is
Yeah.
important. Yeah.
Hmm. Cool. So this we're approaching the end here of what I would like to wanted to discuss with you. So And now, so of course, now we are now working on making ever, I guess, larger models, network models of well, rodent networks, or networks maybe in, yeah, especially in rodents, I guess, maybe in cortex, or other cerebellum, and so on, hippocampus. So what is the, I mean, for you who sort of working on understanding human cortical networks, what is highest on your wish list? Is it sort of new information, or is it you need more computing power, or what is the limiting factor?
Computing power is something, but I think there is, as usual, as I see, not a lack of things to do. Like
Mm,
one of our immediate wish list is to do a multi-layer network, hook layer 2, 3, 4, and 5,
mm,
and be able to ask questions that are driven by interaction over layers.
mm,
And again, obviously, this has been done to some extent, but maybe not so much in human models, and definitely maybe not as rigorously as a very focused effort is going to achieve. So this is one thing. Yeah, all in all, the data is out there and waiting to be used for the right questions. That's
yeah.
what I've seen so
Yes.
far.
I
So
guess also with thousand neurons and thousand multi-compartment neurons, if you have a super duper computer so that you can run the model many times with many parameters, that's always useful in the fitting,
yeah,
like in
yeah,
other.
I think computing is an issue. We've seen it also. Part of our effort right now is to simplify the neurons so
Mm,
that they can run faster, but retaining as much as possible from the complexity of the single neuron.
making it simpler, but not, well, as simple as possible, right?
Yeah,
But
simple as
not simpler.
possible, but also to run fast,
Yeah,
right?
exactly.
Because
Huh. But not
yeah,
and not simpler. So you want to keep that. That's
yeah,
like, of course, the key mantra of of a lot of science, or at least modeling science. So, Itai, I don't know if there was anything with you wanted to say before we close off. Anything I forgot?
no, I think we covered most of the things. I think I'm very happy to, I was happy to discuss all of that. And
Mhm.
again, I'll say again that it's very satisfying to see people using the models, and people I think have been appreciative of the human models as well. And yeah, I think that also exchanging information, ideas.
Mhm.
I think this podcast
Yeah,
is an opportunity for that. So
very
yeah,
good. Well,
that's
that's
great.
one of the privileges, right? That of being
Yeah,
in science, so talking to people all across the world.
yeah.
So we're interested in the same thing. Okay, so thanks a lot.
Thank you.